It's my pleasure to introduce Diane Ramsden to all of you. Diane is an Associate Director in Global DMPK within Takeda. She's currently responsible for representing drug metabolism and pharmacokinetics on program teams and is active in the IQ PBPK induction team and the microphysiological systems affiliate. Prior to joining Takeda, Diane spent 2 years at Alnylam and 16 years at Boehringer Ingelheim, where she served as a DMPK project rep and hepatocyte lab head. She has been a core member of the Induction Working Group since it was established in 2014. Her research interests include the application of in vitro and in vivo models towards developing a mechanistic understanding of the disposition of novel therapeutics. Over to you, Diane.
Oh, thanks very much, Naresh, for the introduction, and Mahalaya and the rest of the IQ Secretariat for organizing this series of webinars. Thanks also to the audience for your interest in the output from the Induction Working Group. As mentioned today, I'm going to be talking about the factors impacting the magnitude of clinical drug-drug interaction due to induction, with a focus on CYP3A substrates. As Mahalaya mentioned, we will make these slides available after the presentation, but I recently learned how to make QR codes, and I'm putting them on everything. So you can also scan the QR code and get the PDF file if you'd like to follow along as I proceed in the presentation. Before I begin, I do have to acknowledge that while this was a group effort within the International Consortium or the IQ group, the presentation does not reflect consensus view of industry in general. Additionally, the views that I express are those of myself and not necessarily my current or prior employers. As a brief background to the Induction Working Group within the IQ Consortium, this effort fell within the Translational and ADME Sciences leg. I'd like to acknowledge all of the contributors over the years to the Induction Working Group. It spans quite a bit of time, and as you can see, contains a number of members, up to 23 core members, and a lot of different companies. And there are 25 different companies along the path that have provided insight and expertise into this work. And many of us have had job changes over the number of years. In fact, I'm currently working for Suresh Balani, so it is definitely a small world. One thing to note is the names that have been highlighted in bold are the contributors to this work that I'm going to be describing today, that we affectionately called the clinical commentary as a short abbreviation as we were going through the writing process. It's important to also acknowledge that we did have expert statistical support throughout all of the work that was completed by the Induction Working Group. And we also pulled in some honorary Induction Working Group members specifically for their insights in terms of this clinical commentary, including Neil Parrott and Sarah Robertson, for their expertise both in PBPK modeling and clinical pharmacology, respectively. I also have to thank the IQ Consortium, which is a not-for-profit organization of pharmaceutical and biotech companies with the mission to advance science and technology in order to augment the capability of member companies and develop transformational solutions that benefit patients, regulators, and the broader research and development community. Before I begin going into the specific details related to the clinical commentary, I did want to provide some background in terms of the Induction Working Group in general. So what I'm showing here is actually what we call the data jam session. This was when people could still get together in person and talk out and discuss ideas, and this really helped enable us to analyze and interpret the data much more quickly. We all set aside a couple of days and really focused on digging into this and into this alone, which was of high benefit. We started the Induction Working Group in 2014 and have sunset it now in 2021 after meeting all of our objectives. The goals really were to provide some recommendations related to induction drug-drug interaction, both the in vitro and also the in vitro to in vivo extrapolation. And we did this by collecting a ton of data, as I'll kind of go through as we move forward in the presentation. We did want to take a deep dive into the variability that we've observed in both the in vitro data as well as in the clinical data. And we've done this previously from the in vitro data in a previous webinar that we hosted a couple of years ago. So today I'm really going to focus more on this end, on what's the potential drivers of this magnitude of variability observed in the clinical outcome. And through that, I'll provide an overview of the clinical commentary. Before I get into some additional background, I did want to highlight that I will be reviewing 6 of the potential contributors to variability. And just to kind of orient you into where I am and what the focus is, I'm going to include this slide as kind of a preface, so you can follow it along the circle and know when we're almost done. So in terms of the induction working group, one of the first objectives was to really understand what the key concerns were for the members in terms of induction regulatory guidance. And the team members agreed to focus primarily on these 5 key concerns. The first, evaluation of downregulation. The second, in vitro assessment of CYP2C induction. The third, the use of CITCO as the positive control for CYP2B6 induction mediated through CAR. Fourth, the data interpretation, particularly this recommendation by the EMA that a 2-fold increase in mRNA is evidence of positive induction. And lastly, which will be covered next week, is the duration of the incubation of hepatocytes with the test article. The IQ induction working group was broken up to facilitate our ability to address these 5 key concerns. So the full working group engaged on recommendations related to downregulation, CYP2C induction, and CYP2B6 positive control. And we did this by surveying member companies and IQ member companies in terms of their current practices and their view on these aspects of induction-mediated drug-drug interaction. The second sub-team was a data analysis team, and this was led by Jane Kenny at Genentech. We really wanted to go into further detail in terms of interpreting the in vitro data and how to context this against a clinical risk assessment. This resulted in the manuscript in 2018 that provided recommendations on data interpretation. What I'll be discussing today is the second output from this data analysis sub-team, and that was really with the intent to be able to begin discussions around the potential contributors of the variability observed in the clinical data that, as discussed in the paper from Jane, really complicated our interpretation of in vitro and vivo extrapolation. The last paper, which was published last year, it includes recommendations on data fitting and optimal time course. And then, as I mentioned, the objectives of the team were met. So the induction working group is sunset in 2021. I just briefly wanted to cover the recommendations just as a reminder of what was decided and determined in the first 2 papers. The objective overall was really just to move towards a better understanding of the regulatory approach to these induction-based drug-drug interactions, and then take a data-driven approach and work with regulatory agencies to hopefully update the requirements for assessing induction And we actually were cited, both of these papers, in the final FDI guidance, which was released in 2020. So in that scenario or regard, I think that our intent was successful. In terms of the overall observations and recommendations from the first paper, where we were really trying to gain understanding around the current industry practice as it related to downregulation, CYP2C induction, and the positive controls for 2B6, as I mentioned, we conducted an industry-wide survey, and then we analyzed the survey. And the survey really led us to a number of observations. In terms of the downregulation aspect, while we had many companies that had observed downregulation in vitro, we didn't have any companies that indicated that this resulted in a clinical manifestation, which in this case of downregulation would probably be consistent with inhibition or an increase in exposure of a sensitive substrate. In culling the literature, we also could not find any examples of clinical relevance to this in vitro effect. So we noted that based on not having the clinical data, it's really difficult to establish an in vitro and in vivo extrapolation. And then, therefore, the in vitro observations really need to be considered with caution. In terms of CYP2C induction, as many are well aware and has been published in literature, there's a really low dynamic range of response in human hepatocytes, which really makes it very difficult to accurately predict the potential for drug-drug interactions. So we collated data from across the industry members, and the observation was consistent with this knowledge already available in the literature. Another observation was that hepatocytes did tend to maintain functional CAR regulatory pathways, making them reliable tools for assessing induction through CAR. We also noted, based on a larger data set, that the likelihood of missing a CAR activator when you're using 3 different donors of human hepatocytes is quite low. In terms of recommendations, we did make some recommendations given the positive correlation between CYP2C induction and CYP3A4, that CYP3A4 can initially be used as a sensitive marker to assess clinical induction. And then after, depending on the outcome of the CYP3A induction-based drug-drug interaction, sponsors could then assess the potential for CYP2C. In the cases where there's a low risk for CYP3A induction based on clinical data, then you can also extend that potential risk to CYP2C. And the last recommendation was that either CITCO or phenobarbital could be used as positive controls to assess CYP2B6 induction in the human hepatocytes. The second paper, led by Jane Kenny, was aimed at data analysis and interpretation, with the primary goal being to better define the in vitro induction positive and negative cutoffs using the collective industry data. And we really were focused on whether twofold change in mRNA levels were appropriate in determining that the molecule was a positive in vitro inducer. And this comes from the EMA drug-drug interaction guideline. There were actually a couple of scenarios where we were, through our data analysis efforts, became aligned with what was recommended in regulatory guidances. One was that you should continue to evaluate induction potential using 3 separate human donors in vitro. And the other was that twofold induction with some concentration dependence is actually a reliable threshold for identifying positive in vitro induction for CYP3A using mRNA level changes. Some new practices also emerged. We evaluated the potential for referencing against the prototypical inducer, rifampin, and being able to characterize or bin the potential for a test article to be a clinically relevant inducer. And our data demonstrated that there was a lack of convergence, and it didn't really account for interdonor variability, which was initially the intent. So it could be misleading to reference against the prototypical inducer. And in some cases, true positives could be missed if you apply a cutoff based on the percent of prototypical inducer response. Another new practice was that in order to kind of reduce the risk of false positives based on the in vitro data, if you don't see concentration dependency, but you do see an increase greater than twofold, this should be observed in more than one donor. And this is really in scenarios where you have that baseline variability, but you don't have clear concentration dependence. If you see that in one donor, then it's safe to say that this is not an in vitro inducer. But if you see it in more than one donor, there might be something to that, and you can maybe redesign in vitro investigations to better characterize that effect. The next new practice was if you are saying that a compound is negative for CYP3A for induction through mRNA levels, you should have at least a 10-fold response in your rifampin positive control. And this is because that dynamic range will provide you an ample window to be able to determine induction risk for those molecules that end up being clinically relevant weak inducers. And the last, which we consider confirmatory, was that the inclusion of a negative control really does not add any value beyond that of the vehicle control. So the focus of this webinar series is the last two papers that were put out by the Induction Working Group. As mentioned, I'm going to be talking about the clinical variability today. And then next week on the 25th of February, Simon will be talking about the recommendations related to data fitting and the time course for in vitro CYP induction. So hopefully you will all join that webinar. Before I get into actual data, I did just want to remind everyone or clarify why we sought to even put this publication out. And as I mentioned, we call it the clinical commentary for short. So basically, we collected a ton of clinical induction drug-drug interaction data, and we touched upon a little bit some of the complications of conducting our in vitro and vivo extrapolation in the paper by Jane, but we didn't really do it justice. So we wanted to make sure that we highlighted areas where this made it complicated, complex. Maybe there were things that could explain some of the range in responses observed, but not really make recommendations, but rather just highlight this as a potential discussion point and something that really requires some further investigation. One thing I do want to note is that a key conclusion from that paper was that simple models could be used despite the variability, both in the in vitro data as well as the clinical data. So we do have confidence in our ability to conduct an in vitro and vivo extrapolation to a certain extent, but there are still some aspects that we need to understand a bit better. So in order to kind of go into the focus of this talk, I just wanted to provide a refresher on what we mean by challenging in vitro and vivo extrapolation. So in order to conduct our IV-IVE, we collected induction data for 51 compounds, and this was initially the in vitro induction data. We then asked, this actually included an ask from individual companies to provide proprietary data. And we were hoping also to get both the clinical and the in vitro data for these proprietary compounds. And with that data request, we actually received 16 compounds, and then the other 35 were gathered from the literature, making up our total of 51 compounds. And then we also collated the clinical data. And if you look at the clinical data by considering the strongest induction, you can see kind of a bell-shaped curve. It's not typical, you have a number of inhibitors, weak to no inducers, pretty high number of moderate inducers and a couple of strong inducers. What was interesting is if you actually look at the same data set, but consider the median clinical response, you really shift towards the left. So we're having more inhibitors, we're having less observations of meaningful induction drug-drug interaction, more mild inducers, and then less of the moderate and strong inducers. So this really made it challenging, and it required us to look at the data in multiple different ways. This is just another view of the type of data that we collected and how we evaluated it. So these each represent individual trials. As you can see, individual trials result in some cases, very different outcomes. What I'm showing in the left-hand graph are those molecules that we considered to be quote-unquote clean inducers and that they don't have any competing mechanisms of drug-drug interaction. And then the right are those inducers that also demonstrate some in vitro inhibition. So you can see a range in magnitude for each of these kind of complex in vitro inducers in terms of the magnitude of response. And you can also see in terms of the breakdown that actually more of the in vitro inducers, 60%, had multiple mechanisms of drug-drug interaction. So it's more likely to have this complicated scenario than to not have this complicated scenario. Another important note before I go to the next slide is that we did actually look at the data in a couple of ways, and one of those was we know that there can be a difference in response dependent on how long the inducer is dosed for. So we only included data that was dosed for greater than 5 days. We also know there's a dose effect. I'm going to touch upon this a little bit. So we only included trials with dose levels that were greater than where you see maximal induction, or in the case of ritonavir, maximal inhibition. So just to reiterate, the focus of this was really to highlight complications of interpreting this clinical induction data. I'm going to talk about many of these, but the variability in baseline levels, the fraction of metabolism, the contributions from other inducible enzymes, the potential impact of intestinal versus hepatic induction, and then the role of study design, including the time to steady state dose level and the evaluated data, all contribute to this range of outcomes that we observed. And I've said this a couple times, but we really did not intend to provide recommendations in this manuscript. We really just wanted to highlight these areas of concern. So this is just a snapshot of data with the intent to really show between clinical studies how variable the outcome can actually be. Even with rifampin, which is our very strong inducer. Sorry, I'm just, there we go. My computer had a goblin in it or something. Oh my goodness. Okay. So just to highlight ritonavir, which can actually range from very strong inhibition to strong induction. And this is really dependent on the object or substrate being evaluated in the clinical study. Some of the speculation around this were that, as mentioned, the basal expression in both the gut and the liver, the study design, the variability in perpetrator exposure between studies, and the lack of availability of this data, and also the selectivity of the substrate. Some of the solutions that we thought about within the paper were maybe there's an opportunity to standardize some of the aspects of this clinical study, including the dosing regimen, dose level, and characterize the perpetrator concentrations during this study. As I'll describe, particularly for rifampin, you can see a huge variability even just in the PK for the perpetrator that can contribute to some of the variability in the magnitude of outcome. Another aspect is that we were focusing our efforts on mean or median data, which was as published in the various publications, and maybe having the availability of individual subject data might be helpful, particularly in kind of including or excluding the variability and number of subjects as contributing contributing to the magnitude of outcome. Another is whether biomarker data might be leveraged earlier, and if PBPK modeling could also be incorporated. So we're back at the contributors to variability, and I think I'm at the point where I'm actually going to go into some of these in a little bit more detail, starting with the baseline CYP3A levels. Before we do that, I did want to remind everyone about how we collected the data, because this is really relevant in understanding each of these contributors and how they might impact the range of responses that we observed. So within the data analysis subgroup, we collected in vitro induction parameters. We did this by asking companies involved whether they had in vitro parameters for common inducers that they're willing to share, either internally or published in the literature. We also sought to identify some additional inducers in the weak range, just for understanding better the translation of weak in vitro induction to a clinical manifestation. We also, as I mentioned, asked member companies to provide their proprietary data, and preferably data that was regulatory submission quality and with clinical drug-drug interaction results. And then we also collected the in vitro induction parameters for the proprietary molecules. And at each of these stages, we actually needed to collect the clinical data as well. The common inducers that we collected all of the clinical data. For the other inducers that we identified, we had to collect the clinical data. For the proprietary compounds, we collected clinical data. So there's a lot of stages here where we're collecting clinical data with the hope that we can rely on this in conjunction with the in vitro derived parameters to evaluate our ability to use in vitro data to predict a clinical risk. In terms of the breakdown of all of this data, we had a 30% from the proprietary information submitted by the IQ member companies. We initially received 17 inducers and one, which was a CYP3A2 inducer. We didn't have enough data to feel comfortable making recommendations for CYP2B6 or CYP3A2, so we really focused our efforts on CYP3A, where we had the majority of data. We requested data for common inducers. That was 28% of our data set. We collected in vitro induction parameters from the literature sources. That was another 30% of our data. And then we generated in vitro induction parameters in 5 of the labs for 5 compounds, and that resulted in 10% of our data set. In terms of the clinical data, we went to the University of Washington database and we searched for clinical AUC or clearance changes that were negative or positive for inhibition and induction. We then searched each of those objects to see whether or not CYP3A contributed to its metabolism, and if it did, we included it in our analysis. In terms of overall output, we had a number of molecules, including those labeled with compound, which are indicating that this is a proprietary compound from a member company, where the clinical manifestation was inhibition. I'm also separating this based on whether all substrates are included or if it's just a sensitive CYP3A substrate. And you can see that the number of clinical studies tends to decrease when we're focused solely on the sensitive CYP3A substrates. We had a number of data sets that fell within a categorization of no induction, and again, a range of in vitro parameters. So each of these is a separate data set. In some cases, we had a huge number of data sets, and then other cases we had fewer. And again, we separated these based on the substrate specificity. In the case of the green star here, this indicates that this was one of the molecules that we identified as potentially weak inducer, and then generated the in vitro induction parameters for. We had a large number of weak inducers, again, with number of in vitro data points and clinical interaction studies. And again, we generated, in some cases, the in vitro induction parameters to help us with our IVIVE assessment. We had a handful of moderate inducers. Again, a number of these were actually submitted as proprietary data from member companies. And we had two that could be considered to be strong inducers, one proprietary and then rifampicin. So all of the data, we had a good range of clinical effects, and we were really seeking to understand better this no effect or weak induction categorization, and this accounted for 63% of the data. So we were able to succeed in building out this data set to really understand the translation of in vitro data to a weak or no clinical induction effect. As I mentioned, we separated these based on all of the substrates available with some component of CYP3A metabolism to those that were considered to be sensitive towards CYP3A. And this is really based on the characterization provided by the FDA of which substrates are sensitive towards CYP3A. And you can see that in many of these categories, based on here the minus sign, that a number of compounds actually fall out of our assessment if we limit to just the sensitive inducers. And in some cases, you see pretty significant induction. So this is kind of the rationale for including all of the substrates, even if their fraction metabolism through CYP3A might be lower than others. Okay, now I'm really going to talk about some of the contributors to variability. I got ahead of myself a little bit, but it's been discussed heavily in the literature, the variability, particularly in the baseline levels of CYP3A. This can be due to both intrinsic and extrinsic factors. There's also some variability between populations, so Asians versus European, women versus men, elderly versus young, healthy volunteers versus patients. There's intra-individual variability, which can be quite high, and even intra-individual, which can be significant. There's also diurnal variations in the activity that have been observed. So there's definitely variability that's contributed based just on the baseline of CYP3A. But there are actually some potential ways to account for this, including orthogonal measures of biomarkers, as noted here, or even with exosome analysis. So while there is variability, there's a potential to learn how to maybe control or account for this. In terms of substrate selectivity, I touched upon this a little bit by saying we allowed all substrates despite the magnitude of or the role of CYP3A in their metabolism. As long as they were metabolized by CYP3A to some extent, we included them. And it can obviously complicate the interpretation of results. So if we look at all substrates and CYP3A sensitive substrates for, in this case, this is primarily those perpetrators which have competing inhibition. And we just look at the max induction versus the minimum induction, you can see a range of responses. Often you can see that there is lower variability in that range if you look at only the CYP3A sensitive substrates. You can reduce that, that variability. And then these are also those inducers that have competing inhibition, which we'll get into a little bit more detail in a few slides. However, there's still high variability between studies with selective substrates, and this is something we wanted to understand further, particularly if you look at rifampin, the variability between using all substrates and using only sensitive CYP3A substrates is pretty consistent. So if we think about how the role of CYP3A might contribute to the outcome in terms of AUCR change, you would expect as the FN CYP3A increases, so does the magnitude of interaction. But the reality actually is that most of our molecules are not just metabolized by CYP3A. They're also metabolized by other enzymes that might be co-regulated, or their distribution or disposition is also mediated through the role of transporters like PGP, which can be co-regulated with CYP3A. So this becomes very complicated. And if you think about trying to tease this out by looking at different inducer and substrate pairs as depicted on this table, you can see that rifampin, highlighted here, is a strong inducer, and this is consistent when this substrate is very sensitive to CYP3A. But then as you decrease the FM towards CYP3A, you can see that rifampin becomes a moderate inducer. However, when both induction and inhibition are contributing to the observed DDI, example being ritonavir, and the outcome of the fraction through CYP3A versus co-regulated proteins actually drives the direction of drug-drug interaction. So with midazolam, sensitive index substrate, you see strong inhibition. With voriconazole, also metabolized through co-regulated enzymes, you see strong induction. This is important when you're trying to assess the potential for clinical induction. We also took those examples from the previously displayed table where there was both midazolam as well as additional substrates evaluated for the same inducers. And you can see that those substrates that maybe aren't metabolized as significantly through CYP3A do not correlate with the change based on midazolam as an index substrate. Whereas those that are very sensitive towards CYP3A, and triazolam is also considered to be index substrate, you can see a good positive correlation. What's important to note is that in some cases, it's the lower sensitivity substrate where you actually see a manifestation of induction. In some cases, the induction is, on the verge of being a strong effect. And you don't see this with the CYP3A selective substrates. Another thing to note is that the actual magnitude of induction can be strong with very sensitive inducers and then only moderate or weak with less sensitive inducers. If we think about the role of FM-CYP3A and compare across substrates, you can see in some cases like triazolam, midazolam, and fentanyl, simvastatin, you see very consistent, strong induction observed. But there are some outliers to this. If you look at lopinavir, which has a high FM-CYP, but a moderate induction, or elaglustat, which has a low FM-CYP, but a strong induction. And across substrates with kind of similar FM-CYP3A or even lower magnitude of response is consistent. So this kind of highlights that the fraction metabolized through CYP3A, while it can explain some of the variability, does not explain all of it. And as I alluded to, there can be a substantial role, especially in the case of competing inhibition and induction, where co-regulated enzymes, which also metabolize the probe, can help contribute to the magnitude of induction. And I gave the example of ritonavir here with voriconazole, but you can see that not only does 3A metabolize voriconazole, but so does the 2Cs, which are inducible through mechanisms related to CYP3A, as are UGTs. So this in the case of sensitive or median substrates may manifest in inhibition, but when you remove the sensitivity of CYP3A, you can see a manifestation of the induction rather than the inhibition. And this is another way that we tried to look at the data to try to conduct our in vitro and vivo assessment. Now I'm going to touch upon how intestinal metabolism and intestinal induction may contribute to the overall magnitude of effect and the variability observed between clinical studies. So we had a number of examples where midazolam was dosed both IV and oral, where the IV would give us an understanding of the hepatic contribution, and then oral would be a combination of both hepatic and intestinal contribution. And you can see for rifampin, the strong inducer, you can see a difference in the overall magnitude of induction, depending on whether it was the object was dosed IV or oral. In other cases, that pronounced difference is not reproducible. So with efavirenz, a moderate inducer, you see some effect, but it's not as dramatic as what you're seeing with rifampin. St. John's wort is a little bit of a special case because the variability in clinical data, as you can see, can be quite high. And this has been shown to be due to the hyperforin content and the differences between formulation in these studies. But there's still, in some cases, a change in oral administration versus IV of the object, but it's not, again, as pronounced as midazolam. And with flecaneril, there's really not much difference at all between IV or oral midazolam. Okay, so in order to try to tease out the role of the fraction metabolism through CYP3A and then the intestinal component, we did some simulations using the mechanistic static model. And because we're focused on induction, we kind of removed the consideration of inhibition and time-dependent inhibition just for the purpose of the simulation. We also had to make an assumption that the hepatic induction parameters that we generate in vitro using hepatocytes will directly translate to intestine. And there's a lot more work needed to really confirm whether this is a true scenario. But in putting varying FMs and FGs into this mechanistic model, you're able to kind of simulate what effect you would anticipate based on those parameters. So taking the in vitro parameters for a strong inducer like rifampin, you can see that the strong induction is always manifested if the FG is or the fraction escaping the gut extraction is low, up to 0.7. It's only when the fraction escaping gut metabolism is a bit higher that FM becomes more important in terms of determining the magnitude of induction. And I have to thank Jessica Remmel from Eli Lilly because she actually showed me this, and I thought it was a really nice way to kind of describe these kind of contributing mechanisms and the potential role they might play in the magnitude of outcome. So one of the responses actually when we submitted the paper the first time was that we didn't have enough examples of IV and oral administration to kind of make strong conclusions related to the role of intestine versus hepatic. So we were asked to identify whether there were some additional examples available. So we did that. We looked again through the clinical data. We looked for IV and oral administration of substrates, and we also tried to get a range of substrates that would be consistent with low to high extraction ratios. We then estimated the fraction escaping intestinal extraction and the hepatic extraction from the reported IV clearance data. And we did this by using equations which have been described in work by Evan Karash on alfentanil. We also used the hepatic plasma blood flow reported in that manuscript. We are presenting the equations here, and then we compared actually the hepatic extraction and the the gut extraction in the presence and absence of an inducer. So if these were similar, then we would conclude that there's really no difference between the hepatic and intestinal induction in terms of their contributions to the magnitude observed. If it was greater for the hepatic portion, then you would see a higher effect on the hepatic extraction than the intestinal extraction. And if it was higher in gut, then you could say there's a higher effect on the intestinal extraction compared to the hepatic extraction. We were able to identify data that was available for 8 of these CYP3A substrates and went through the process of calculating these parameters. And in most cases, you can say that the hepatic and intestinal induction is similar. There are, however, some examples where hepatic induction exceeds that of the intestinal induction and vice versa, where gut is greater than hepatic induction. So if we look back at the table I showed previously when we were talking about FM CYP3A, you can see again that FM CYP3A is not the only contributor. And then if you start to tease out what role FG might have in this magnitude of interaction, you can see when these two parameters are similar, you see similar response. When they differ, you can start to see a different response. The example is eliglustat, where the FM-CYP is quite low, but so is the FG. So the impact of rifampin is actually strong induction. Another consideration in looking at this data is that when there's actual role of OATP substrates, rifampin's an inhibitor of OATP, so you can actually see an impact on the magnitude of induction dependent on the study design. So there is definitely a special consideration in considering the potential interactions when you have, as well as contribution from CYP3A, some active hepatic uptake. I'm now going to touch upon the impact that inducer PK, including the dose and the regimen, might have in the overall magnitude of interaction. So we were able to collect some dose response data. There's a lot of clinical data for midazolam, and there's titrations with rifampin. There's similar data set for alfentanil. So you can actually generate a dose response curve and clinical induction parameters based on this curve. And you can see the differentiation between IV and oral for the midazolam data. We also collected multiple doses for other objects. And in some cases, like with rifabutin and oxcarbazepine, you can see there's really not a change in the magnitude of induction with the increase in dose level, as you see with many of the others. And this might just be because you've already reached the maximum induction at the lowest dose level that was tested. Another thing to highlight is that the slopes of these dose response curves differ across the molecules, and this might be driven by their potency, which you can also see in the in vitro data when you estimate those induction kinetic parameters. A point of caution is that often when we assess the clinical likelihood of induction, it's reasonable and required to test the top maximal dose that you might be expected to be efficacious, but it might not be from the perspective of assessing induction potential where you would see your maximal effect. So it's just important, a note of caution when trying to look at data across multiple clinical studies that often the Emax may not have been achieved. If we think about this study duration, there was a lot of data where rifampin as a strong inducer was dosed for a number of days, and then the object PK was assessed. So you can see even within day 5, maximal induction by rifampin for many of these objects and substrates. So days 5, 6, 7 tend to be consistent with the magnitude observed, at later time points like day 14 and day 28. A notable exception of this is caffeine. Caffeine actually shows a stronger induction later than what's observed at day 7. And this kind of highlights the differences potentially in the time course for induction needed, depending on which enzymes are contributing to the metabolism. So caffeine being a 1A2 probe might explain the delay in terms of achieving maximal induction. If we think about whether this translates across potency classes, so moderate inducers such as basantin and phenytoin, you can again see that the response at day 7 or day 5 is similar to the response at later time points. If we then look at no induction or weak induction, again, the trends are consistent. You can see maximal induction by day 5, day 7, across weak inducers as well. So taken together, the strong, moderate, and weak inducers suggest that for CYP3A induction, a dosing regimen of 5 to 7 days is sufficient to achieve maximal induction, but only if the half-life of the inducer is similarly short to the half-life of CYP3A. That being said, there is a nice paper out which uses PBPK modeling and tries to tease out the intestinal and hepatic contributions. And this is also due to the variable responses that can be observed with rifampin. And they recommended that rifampin be administered for at least 10 days. And this is because in their modeling and simulation, it takes longer for hepatic induction to reach its maximal effect. And this really highlights a need for further investigation into this before recommendations can be made to modify any clinical study designs. Okay, so there are some complications with rifampin which have been highlighted in other papers. Primarily this variability. As you can see, when you look at all substrates, the similar extent of variability is observed as when you look at just CYP3A sensitive substrates. And part of this discussion is what is different or potentially different, or how does the difference in variability in the rifampin PK contribute to some of this change or variable observed magnitude of response. One thing to note is that often we don't look at the perpetrator PK, so it's difficult to say how it might contribute to the variability. In terms of rifampin, we know it's inducing a lot of drug metabolizing enzymes and transporters. There's also some crosstalk with between nuclear receptors. There can be pretty high variability in terms of inter-individual difference in PK, and this may be due to polymorphisms in either the transporters that contribute to its disposition or the metabolizing enzymes. It's known to have time-dependent PK because of some autoinduction observed, and it's also demonstrates greater than dose proportional PK due to its extensive saturable first-pass metabolism. And in terms of assessing magnitude of induction, I think in some ways it would be impactful to capture the PK if it might be a contributor to potential variable responses in terms of clinical outcome. I'm now going to touch upon how transporters and their co-regulation with CYP3A or inhibition of these transporters can contribute to a variable outcome in terms of magnitude. So we know that transporters are important in mediating disposition often of multiple substrates. And in terms of PGP, which is co-regulated with CYP3A, there is broad overlap. When you see induction of CYP3A, you're also likely to see induction of PGP, albeit into a different like bin. So a strong inducer of CYP3A may be a moderate inducer of PGP. And then if in the case where you have a co-substrate of PGP and CYP3A, there's some evidence that you can underestimate the observation of or magnitude of rifampin induction if you don't incorporate that PGP component into the evaluation. In terms of hepatic uptake and efflux transporters, if the inducer is a substrate, it's been shown, transporters can modulate the intracellular inducer concentration, and this can affect the magnitude of effect. And when the substrate is also a substrate for transporters, such as repaglinide and maraviroc, you can see an underestimation of the magnitude of induction if you co-dose rather than stagger the doses when you're assessing the object's PK. So an example, some examples of this is in terms of PGP and the impact on intestinal induction. You can see a variable outcome in terms of the role of intestinal versus hepatic induction. In the case of these quinidine and verapamil, both of these are substrates of PGP. In addition, rifampin has been shown to inhibit PGP. An example of where hepatic uptake can contribute to variability in the magnitude of response is shown for repaglinide, which is also a substrate for OATPs. And this, if you co-dose, you see a moderate induction versus if you dose the repaglinide after you've administered rifampin, you can see a more pronounced induction effect. And this is because rifampin is an inhibitor, or has been shown partially to be due to rifampin inhibition of OATP-mediated uptake of repaglinide, which underestimated the magnitude of induction. The last thing I want to touch upon is complex DDI, which is an important consideration because, as I mentioned, we see a lot of inhibitors that a lot of inducers rather that are also in vitro inhibitors. If we look at the data set that we collected as part of the induction working group, we see 61% of our inducers also demonstrate some inhibition. As I've already mentioned, if you look at only sensitive CYP3A substrates, you actually miss the opportunity to evaluate in vitro and vivo extrapolation of 25% of the compounds we collected in vitro induction data on. So we had a data set that had a large portion of both CYP3A inducers and inhibitors. And one of the main questions in this case is, okay, what's gonna happen? And I have to thank Tom Chan, a former colleague at BI, who I took this graphic from because I really like it. I think it's a nice way to illustrate that, in the case of induction or inhibition, it's going to be really dependent on those in vitro parameters, so like that, what is going to be the outcome? Is it going to be no effect? Is it going to be inhibition? Or is it going to be an induction effect? And you can see this, again, same table that I've shown a couple of times now in the interaction when you look at these co-inducer inhibitors and compare their median effect versus the worst case. And this is really driven by co-regulation of other enzymes metabolizing the probe. And then this becomes an important consideration for assessing the potential for clinical induction. The last thing I'm going to touch upon is the special consideration regarding oral contraceptives. So we were able to collect clinical data for 23 inducers where there was interaction data for ethinyl estradiol, levonorgestrel, and norethindrone, as well as a sensitive CYP3A substrate to kind of compare the magnitude of effects between them. 4 of these shown here showed no induction or weak inhibition. 5 shown here showed some differential effect dependent on the substrate, This ranged from inhibition to weak induction. In this case, aprepitant, ritonavir, perampanel, and brivaracetam were all identified as weak inducers with oral contraceptives, whereas with sensitive substrates, they didn't show induction effect. We also look to see what this may manifest depending on the binning, whether it's a weak, moderate, or strong inducer. In some cases, you could see similar magnitude of effect between these oral contraceptives and sensitive CYP3A substrates. Examples being nevirapine, rifabutin, and troglitazone. And in some cases, like with oxcarbazepine, you actually see a higher induction with these than with the sensitive CYP3A substrate, although the binning is still a weak inducer. In the case of strong inducers, such as carbamazepine, phenytoin, and rifampin, you can see that the magnitude of response with the sensitive substrate or index substrate is higher than what is observed with the oral contraceptives, which are binned in a lower categorization in terms of overall magnitude of response. And so some of the important things to consider when we think about clinical induction assessment, particularly for oral contraceptives, are that there are multiple enzymes, including those that may be co-regulated with CYP3A, which contribute to the metabolism of them. And this is published in a paper in 2018. Since the inducers of CYP3A can also induce other enzymes involved in the metabolism of oral contraceptives, understanding the relative role is important, particularly in weighing the need to conduct a clinical drug-drug interaction evaluation. Of note, evaluation with oral contraceptives in drug-drug interaction studies can appropriately identify CYP3A inducers. The magnitude on other substrates is difficult to extrapolate, and PBPK modeling may help to understand this complex drug-drug interaction. But in the case particularly where there's competing mechanisms, there is some precedent that rather than inhibition, you can see a weak induction. So that leads me to my last topic, which is just the potential for PBPK modeling, which takes into consideration the physiology of the human body and in assessing the potential for drug-drug interaction. When we think about the data analysis that we did in the data analysis paper published in 2018, we weren't set up to do PBPK modeling, particularly for the proprietary compounds, because we didn't have all of the input parameters needed. But we did look at a number of variations of these static models. And we determined that when you use the average concentration, you get the best quantitative prediction. So this really kind of highlights the potential for exploring additional means for assessing quantitative risk of induction-mediated drug-drug interaction, including PBPK. There is a new working group focused on this effort within the Translational ADME Leadership Group. And the goals of the project are really to try to build the data set out to improve the likelihood of acceptance by regulatory agencies who have historically shown some reluctancy, primarily because there's not enough examples available in the literature. So they really like to see further validation with a larger data set. So the goals of this group are really to generate those additional examples, including assessing a translation across induction categories, weak, moderate, and strong, and then establish some best practices within the industry. So the initial primary objectives are to establish the baseline situation with a survey, which went out recently. Establish best practice in terms of how PBPK can be approached, particularly in the case of assessing drug-drug interaction potential through inducers by building out the number of published examples and also the experience within the working group. And then we're going to actually do some modeling, both for existing models and establishing new models, to try to build confidence in this area. I'd like to again acknowledge the IQ Consortium for the support, my colleagues within the IQ Induction Working Group. I'd like to remind everyone again that this is just part one of the two-part series. The second part will be presented next week by Simon Wong, and I encourage you to join that as well. And lastly, I'd like to thank everyone for their attention, and I'm really happy to start to take some questions. And again, if you'd like to a copy as well, if you want to download that.
Excellent. Thank you so much, Diane. That was an excellent overview of manuscript 3, the variability part, the clinical commentary specifically. There are some really good questions in the chat. So I'm going to start with the first one. To what extent are the different methods used within companies responsible for the variability? What would you expect if the same methods were applied by everyone?
Yeah, so this is really interesting, and it is something that we attempted to understand as part of the data analysis efforts, where we collected multiple parameters from different companies that employ different conditions, particularly in terms of the in vitro assay. And then we tried to apply some statistical methods to look at the impact of those in vitro study, the differences in the in vitro study design, and how they might contribute at least to the variability observed in the estimated induction parameters from those in vitro data sets. And unfortunately, that type of analysis requires quite a big sample size, which we didn't have. We weren't powered to adequately assess this. So we couldn't dig further into how these things, like in terms of media formulation, or if someone's using sandwich culture, hepatocytes versus monolayer, or if there's protein present in the media, we couldn't really identify specific contributors. However, when we began to analyze the data sets from within companies, there were actually a number of companies that did have repeat studies within the same donor, within the same operator, the same conditions, they're following the same protocol. And the variability observed in that data set was very similarly high to the variability observed when we combined all of the data from the various companies. This data is actually published in the paper from Jane. So I think it's an interesting topic, but based on at least the in vitro data, I don't think it's having a standardized method or approach is going to alleviate some of that variability that was observed.
Excellent. All right, I'm paraphrasing this next question. With variations in baseline values, example mRNA, due to differences in basal constitutive expression, are these differences not expected? And then the variability is a function of the variation in the denominator. Hope that's clear, Dan.
Yeah, thank you. Yeah, I think this is an interesting topic. There's definitely some nuances to this. It's kind of inconsistent, though there is a trend where you see like a lower initial baseline, you can see maybe a higher magnitude of induction. But as I mentioned, it's really not consistent across either in vitro or in vivo studies. There's also, the need to consider those intrinsic and extrinsic factors which might contribute to this baseline, and the variability that comes from the sample size. So like you can decrease the extent of variability with the higher N that you incorporate. It's also possible that if we think about the fold change as like a binned outcome, like weak, moderate, or strong, and not differentiate based on absolute AUC changes, then the variability becomes less of an important aspect. I think it's worth considering whether the magnitude of change might be in some ways more informative if it's based on the intrinsic clearance rather than a fold change in activity, given this aspect of, your denominator might be higher or lower, and that's going to contribute to the outcome. I definitely think that we don't have all of the answers here. And I just want to reiterate that it was kind of surprising, I think, for many of us when we collected this data, there was still so much variability in the clinical data. Even using the same substrate, using the same inducer, using a very similar regimen. So this is something definitely worth further exploration and to see if there might be ways that this could be controlled or accounted for.
Okay, great. This question comes from Swapan Chowdhury. So you showed, again, I'm paraphrasing. So you showed compounds which had inhibition, so competitive inhibition, time-dependent inhibition, as well as induction. How would you dissect the effects?
I think this is a really an interesting question. There's definitely been some proposed ways to do this. There's the mechanistic static model, which tends to overpredict, and then there's PBPK modeling, which, the more complicated it becomes, the fewer examples that there are, the less likely it's to be accepted by the FDA or regulatory agencies in general. So I think the strategy that is put forth now in terms of trying to assess these individual components separately makes sense, particularly in cases where you do see that the overriding mechanism can be different depending on the molecule that's being tested. So I would say that there's still a lot of room for improvement in trying to incorporate these aspects together. One thing that I have heard from other researchers is trying to look at different endpoints. So right now we're taking an mRNA endpoint from induction and we're trying to compare it relative to a enzyme activity measurement of a probe substrate in a clinical evaluation. So like how do you expect an mRNA endpoint, which can be variable and of much higher magnitude, compared with enzyme activity to translate to an enzyme activity endpoint in a clinical measurement? So there are people that are exploring other means, such as protein levels or even more physiological appropriate hepatocyte models, such as the work that Naresh's lab did looking at hepatopac as maybe a way to reduce some of that in vitro variability and get at a reasonable magnitude of change. So there's wrap a long answer up short, hopefully. There are methods, they're not perfect, and they do need some improvement.
All right, great. This next question comes from Eva Hune. How well do in vitro PXR data predict CYP3A for induction?
So we didn't look specifically within the induction working group at PXR data. We were really intent on focusing on the data that is currently accepted by regulators as a means to kind of stimulate discussion within the regulatory agencies. So it wasn't part of our assessment as the induction working group, but I can say that I have seen data indicating that there's a correlation. And often it is important to establish the mechanism. So in a discovery setting, the PXR data is still rather useful. It's high throughput, you can rank order compounds, it's reproducible. But in terms of looking at the big picture, I think currently the gold standard is to use the human hepatocyte model. So that was our focus.
Fantastic. So the next question comes from Odette Farmi. She starts off by saying great presentation. So congrats again, Diane. Just curious to know if you guys have looked at variability in vivo with the different lots somehow is also shown in different subjects.
Yeah, so I think I think hi Odette, it's great to see you virtually. I hope you're doing well. Odette worked on some of this work with us as part of the induction working group. I think what you're asking is whether there's some data available where somebody has looked at the response in vitro compared to the same donor in vivo, and not that I'm aware of. I think that would be really complicated because most of our cell fractions come from, unfortunately, like resections or people that are deceased and their liver can't be transplanted into another person, so it's used for scientific purposes. I think there might be an opportunity to further explore this with the emerging exosome analysis technology. There's some data indicating that the enzymes present in exosomes, particularly CYP3A, are still active. So that might be a way to kind of get at this question. But yeah, I haven't seen, and it certainly wasn't part of the initiatives within the induction working group to kind of try to compare between that. Another thing to note, I think, is even in clinical interaction studies, it's not often that you assess the drug-drug interaction in the same group on multiple occasions. It's usually kind of one and done, and then you have your answer.
All right, fantastic. Next question comes from Sandra Baldwin. Was UGT induction discussed as a potential knowledge gap as to magnitude of risk or not?
Yes, it was, absolutely. We actually discussed multiple potential enzymes that would be of interest or potential importance in terms of clinical manifestation. The 2Cs are one example of this. The 2Cs had enough clinical data to kind of try to go about making recommendations, as I kind of covered. These are reported in Naresh's paper from 2017. In terms of other enzymes and transporters of interest, there's definitely clinical data demonstrating a potential. But in terms of teasing this out, in comparison to like CYP3A as an example, there really isn't enough clinical data available to make kind of firm recommendations based on the in vitro observations. I do know that in some cases there have been labs where UGT or glucuronidation is an important pathway. And in those situations, definitely consideration of a potential for an impact with an inducer should be part of kind of the overall risk assessment as the project's progressing through development.
All right, great. Next question is from Christian Lupfert. Again, thank you for the great talk. Could you expand on rifampicin being CYP3A inducer, PGP inducer, and PGP inhibitor? How can effects on CYP3A and PGP be differentiated?
That's a little bit of a hard question, but that's okay. Yes, it's known rifampin actually induces a ton of things. That's why it manifests in like the strongest interaction that you can observe kind of within inducers, and why it's recommended as a strong inducer. Well, up until recently it was recommended as a strong inducer to kind of leverage your data against in terms of clinical interaction. So for expanding a little bit on the co-regulation of CYP3A and PGP, there are some published examples in the PBPK realm where if it's from Venkatesh Reddy, and you can actually see an underprediction of rifampin. So when you think about the PBPK model for rifampin, when it doesn't consider that rifampin also can induce PGP, you kind of underpredict the extent of interaction, and this has kind of been postulated to be due to that concordant interaction that you might observe, particularly in intestine, where you'd see an increase in PGP kind of kicking your molecule back to CYP3A to metabolize it. So that's one aspect. And then in terms of inhibition of rifampin, I think that's a little bit more difficult to tease out because of that potential for induction. But there are data showing some clinical relevance to rifampin as an inhibitor of PGP. And I think, Naresh, I might have covered each of those points,
You
but if
did.
there's something I missed, okay.
All right, fantastic. Next question. Can you speak to the limitations with applying orthogonal biomarkers to assess baseline differences in CYP3A or magnitude of DDI?
Sure. So there is some language in the clinical drug-drug interaction guidelines where biomarker data is indicated as maybe useful information to collect, but it's not recommended, at least as for index studies, because there's really no consistent, clear translation or ability to extrapolate to other substrates. The couple that I kind of let me try to go to the slide that I mentioned here, such as the 6-beta hydroxycortisol, that's been around for a while, and it's highly variable. There's some recommendation to use the ratio of the 6-beta hydroxycortisol to cortisol to reduce some of that variability. That's thought to be really because of some diurnal changes. But the variability of this is still quite a bit higher than some of the others that have been proposed. There's also a need to collect urine for a longer period of time, which might be difficult in the clinical interaction studies. For beta hydroxycholesterol, there are some data which demonstrate its utility, particularly when combined with other data. So there's a study with midstorin and PBPK modeling where midstorin undergoes really complicated drug-drug interaction, and it includes some interactions mediated by its metabolites. And that biomarker data was actually helped used to build confidence in the PBPK modeling. So that's, I think, an example where it's been valuable, but there's still a concern with the 4-beta hydroxycholesterol due to the half-life being so long, around 17 days. It's also difficult to reproduce results correlating the biomarker change with that of like CYP3A activity measured with like midazolam, and the dynamic response tends to be quite a bit lower. So it wouldn't be helpful in kind of identifying maybe a weak inducer or moderate inducer. I mentioned exosome analysis. There's some recent proof of concept studies here which seem to be really promising, but obviously need to be developed further. And so there's promise there and there's potential, but it still needs to be evaluated. There can be some there's a need to kind of differentiate the origin of the organ. And there's also some indication that you need to account for differences in the shedding factor that can complicate its interpretation in terms of accounting for this baseline variability.
Okay. One last question. You showed evidence that the magnitude for induction is often maximal after day 5 or 7, even for weak and moderate inducers. Does this data indicate that a week of inducer, week as in days of, 7 days, I guess, of inducer treatment should be adequate to achieve maximal clinical effect?
To be honest, I think there's still opportunity to understand the time course for clinical induction better. And this includes the translation across enzymes and transporters, particularly when there's a combination that might contribute, like what we observed with the caffeine for CYP1A2. And so we did make a combined effort to try to understand this aspect from the in vitro time course. So shameless plug, Simon will talk about this next week, I think. You can kind of hear how we made strides from the in vitro end, but I would say we're not there yet. There still needs to be a little bit more evaluation from the clinical end. So I would say it would be difficult to make that recommendation based on the current data and the other data available in the literature.
This question just came in from Dave Stresser. He says, great talk as well. Does the working group see